So, hello everyone, thanks for attending this session. My name is Pedro Echave and today I will be talking about the solution developed by PerkinElmer for COVID sequencing, the NEXTFLEX Variant SARS-CoV-2 kit. So the emergence of mutant SARS-CoV-2 variants associated with changes to transmission and health outcomes have shown that tracking and identification of virus variants is critical to establish an effective public health response. As a consequence of this, once a sample is detected as positive for COVID-19 using an RT-PCR assay, labs are more and more encouraged to find whether this particular sample contains a variant of concern or not. To do so, 3 steps are necessary: library preparation, where the sample is made ready for sequencing, the sequencing itself, where the data is generated, and finally, data analysis. We will review in the next slides how each of these steps are handled by our solution in detail. So let's focus first on the library preparation. The NEXTFLEX Variant-Seq has been designed to make COVID sequencing easy. The kit is available in 96 and 384 reaction format, and it's an all-inclusive solution, meaning with this that all the components that you might need to prepare samples for sequencing are there: the reagents, the barcodes, and the magnetic bits. Also, software is part of the product. We will talk more about this later. So now let's go a little bit into the details of the workflow. As you will imagine, the sample, so the input for the NEXTFLEX Variant-Seq SARS-CoV-2 kit, is extracted genetic material from a swab or saliva, same that was used for your RT-PCR assay. So we'll take this extracted material, and then first of all, the workflow will convert the RNA contained in it into DNA. This is standard in NGS workflows for RNA. Afterwards, the viral DNA will be amplified using the RT-Pull primers that cover the whole genome of SARS-CoV-2 and have been thoroughly tested by the community. Finally, those amplicons will be processed using a standard library prep. This workflow has a total turnaround time of about 7 hours. As we just mentioned, the input material is the same extract used for RT-qPCR assay, and there is no need to do surface extraction or any dilution. No manipulation of any kind is required. Samples with different viral load can be used, and we know that this will have no impact on variant identification performance. In fact, we've been validating our kit with the real clinical samples, and we know that most clinical samples that test positive will have a viral load that is compatible, so it's within the range of our solution. So COVID sequencing is an application where many samples are processed. In this case, automation is relevant, and then we thought of just showing a quick view at the automation solutions we offer. So at PerkinElmer, we have our own automated liquid handlers for NGS, and we have plenty of experience in this field. Our NEXTFLEX Variant-Seq kit can be used manually and also can be automated using our instruments such as Cyclone G3 NGS, Cyclone IQ, and Sapphire. So we have all the scripts readily available, and then different proposals for automated workflow depending on the throughput of the laboratory. So we have solutions or proposals for 96 samples per day, up to more than 700 per day. Don't forget that in addition to the instruments, we can provide also the consumables for automation. So basically, working with PerkinElmer, you can have a single supplier for all your needs for COVID sequencing. Now let's review briefly the part of the process corresponding to the sequencing. First of all, we should say that our solution is compatible with any Illumina sequencer, from an iSeq to an OverSeq. So this our kit is quite flexible, and different sequencing settings are possible. However, we recommend single-length sequencing with a read length of 36 nucleotides, basically to have a quick turnaround time. In all cases, so regardless of the sequencing settings that you use, a minimum of 1 million clusters per sample is recommended. So just to illustrate the relevance of the sequencing settings in time of sequencing, please have a look in to the plot on the top of the slide to see qualitatively how sequencing time is impacted by doing parent or or by increasing the read length. For example, on the table below, you can see that on an NextSeq instrument, using our recommended settings, you can obtain data in less than 11 hours, whereas most solutions in the market are recommending 2 by 150, which has a sequencing time of about 29 hours. So for an application where quick response time is crucial, this is a factor, quite important factor to have in mind. Finally, in the table presented in this slide, you can see the clusters generated by each instrument. These numbers are equivalent to the maximum number of samples that you can load per run if you follow our recommendation of 1 million clusters per sample, as we discussed previously. So in an ISeq, you can sequence up to 4 samples. NextSeq, which is probably one of the most used instruments for COVID application, you can sequence between 130 and 400 samples. And then with NovaSeq, due to the high multiplexing capabilities of our barcodes, then you can sequence more than 10,000 samples per run using an S4 flow cell. So now let's talk briefly about the analysis part of the process. So customers using the NextFLEX Variant-Seq SARS-CoV-2 kit have access to free data analysis. The solution we offer is a web-based application, not requiring any installation, and is really easy to use. Just in 2 steps, customers will be able to drop their FASTQ file, so the raw data, and then analysis will start automatically. Nothing else is needed. The software performs a number of steps, and we wanted just to go over them for you to have an understanding of what it's doing. On the first place, data is mapped against the Wuhan reference strain, globally used as reference for SARS-CoV-2. Then a QC is applied to the data, so to ensure that at least 90 out of 98 amplicons generated by the Arctic primers are fully covered. This is crucial to ensure that we are not missing any mutation. Then the actual mutations are called, and finally the variant is assigned. In this slide, you can see an example of a real report generated by the software. This is a real clinical sample, as I was telling. So the most important part of the report is the summary. Under this section, you will have the main results of the test. You will have sequencing metrics, such as the percentage of base cover. And then also you can see the Pangolin lineage, which corresponds to the variant that we want to determine. If the variant is considered as variant of concern, this lineage will be flagged as red color. After this summary, you have an additional table with all the changes detected in the sample with respect to the reference genome. Also familiar with this application, so Pangolin is the standard tool used for SARS-CoV-2 lineage classification and nomenclature. Here you have in the slide a link to the website where you can visit. Then you will find that in addition to be a tool for classifying your data, it also contains a lot of information on major lineages and epidemiology that is publicly available. I wanted to mention that in this report, in addition to the Pangolin lineage information, there is also information based on NextClass Classification. This is a different open source algorithm for SARS-CoV-2 classification that is also widely used. And as I was telling, it's also included in the report. And then this is all that I wanted to discuss with you today. Just a brief introduction to our solution for COVID sequencing. Thanks for your attention, and I'll be happy to take any questions you might have.